Well, good morning, AP Bio students. Today I'm just recording this. I'm going to record the Screencastify for those of you that need to head out and pick up your textbooks today and are going to be missing today's lecture. So juniors, this one is for you. This is a mini lecture on just protein structure and function, and then also nucleic acid structure and function. So this is the presentation that you guys do have access to on Schoology. And then also for today's activity, if you don't need to listen to me, you can watch the videos that I linked onto the document that you will be submitting as well. So for Unit 1, as we look through, remember that for the unit at a glance, we've already talked about the structure of water and hydrogen bonding. And then yesterday, for all of these, 1.2 down to 1.5, we talked about carbohydrates and lipids. So today, we're going to be talking about how this applies then to proteins and nucleic acids. And 1.6 is then specifically nucleic acid structure that we need to make sure that we're addressing. So as we flip through what we've already discussed so far, here's kind of the flashback of everything. Remember that in organic molecules, carbon is the core of all organic molecules, and in our macromolecules, there's no exception. So carbon makes 4 bonds, hydrogen makes 1 bond. We're always going to find carbon and hydrogen. But in the carbohydrates and lipids, we just had carbon, hydrogen, and oxygen, where oxygen makes 2 bonds. So we're talking today about larger molecules. We're going to talk about proteins first. Protein actually includes all of these elements. So carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. And then when we talk about our nucleic acids, we're going to have carbon, hydrogen, nitrogen, oxygen, and phosphorus. There's no sulfur in nucleic acid. That's going to be an important piece of another unit when we talk about the discovery of DNA. You're going to need to make sure that you know that piece. Now, remember that all macromolecules are going to link their monomers together using dehydration synthesis, and all macromolecules are going to separate their monomers through hydrolysis. So we need to make sure that we are understanding monomers and polymers, and we can recognize then pictures of the monomers. So today we're going to talk about proteins, so you need to be able to recognize amino acid structure. And then with proteins, you're going to need to know the 4 levels of protein structure as well. And then nucleic acids, you're going to need to be able to recognize a nucleotide and then be able to recognize the differences that exist between RNA nucleotides and then DNA and then the molecule structure. Here's our carbohydrates, there's our lipids. So that was the worksheet that you guys worked on yesterday to summarize carbs and lipids. So today we're talking about proteins. And what I always say is that protein function is everything. It really is. From structure to signal messengers to receptors on cell surfaces, enzymes that actually control all chemical reactions, to antibodies, to transport across the cell membrane, to movement. I mean, proteins do everything. If you think about your DNA is instructions for making proteins. So it's actually proteins that regulate the initiation of DNA even being read for transcription and translation too. So proteins really control you, and that's the difference between you and I is the proteins that our body creates, not necessarily the DNA, but the proteins that are produced. So when we talk about proteins, again, I told you that for proteins, you are going to have to recognize primary structure, secondary structure, tertiary structure, and a quaternary structure of a protein and the difference between those. But the first thing we need to make sure that we're clear on is the basic monomer of a protein. Basic monomer of a protein is an amino acid. Now, an amino acid looks like this over here. There's an amino group, and we're going to break this down to our different functional groups when we start focusing on our specific chapters. Your amino group is a nitrogen group. There's a carbon core, and then there's a carboxyl group on the other side. Well, this is really important to understand that there's this carbon core molecule of all amino acids, carboxyl group and amino group, because what ends up happening in forming a polypeptide, when amino acids link to form a polymer, is the carboxyl group is going to bond with the amino group. So what's going to happen is a water is going to be removed because it's a dehydration synthesis reaction. And so it's through the carboxyl, then with the amino group, where peptide bonds are formed. So this is also why our proteins are called polypeptides, because the name of this bond between two amino acids is called a peptide bond. And so if we've got lots of those peptide bonds, it's now a polypeptide. Now, with amino acids, what you need to know is that with amino acids, all of them are going to have this basic structure of a central carbon, an amino group on one side, a carboxyl group on the other, a hydrogen to satisfy bonding rules, and then there's going to be this R group. This is our side chain R group, and this is the variable part of amino acids. Now, there are 20 different amino acids. Now, with those 20 different amino acids, well, that's what gives each amino acid different chemical properties that ultimately leads to the shape of a polypeptide, because these side chain interactions in solution or with each other end up contributing to the folding and the permanent structure of a protein. And it's not permanent because the protein structure changes with pH change and temperature change. But we have to recognize that when we talk about our different amino acids, it's the R group or the side chain that ends up being different. Now, you do not have to ever memorize in AP biology which ones are nonpolar, which side chains are polar, which ones have a charge, a negative charge, which ones have a positive charge. That is nothing. That is beyond what College Board ever wants you to know. They do need you to know basic amino acid structure, and there's 20 amino acids, and these side chains then vary in their chemical properties. That's it. So kind of leave it at that. Now, do remember that when we talk about our DNA, you guys did this back in freshman biology. You guys learned to take DNA's instructions for making proteins. You learned that the code in DNA is written in a triplet code. And what ends up happening is DNA copies itself in a messenger RNA. And then from messenger RNA, you guys probably took the universal genetic code and you read then that AUG then codes for this amino acid. And then AUC codes for this amino acid, and UCG codes for this amino acid. So why I'm showing you this, trying to put some relationship to it, is when we do this and we figure out the order of amino acids, this is what is called the primary structure of a protein. The primary structure of a protein is just that sequence of amino acids. So if we were doing any DNA decoding and trying to figure out what the protein is, all we can actually figure out is what that primary structure is. So in a cell, however, and we'll talk more about the endomembranous system and in the ER, the rough ER, well, now there's enzymes that are actually going to help fold this protein into its proper shape, because the shape matters. Shape is what gives a protein its function. So after the primary protein structure, what ends up happening is this long chain of amino acids, this long polypeptide, gets folded, and it gets folded based on its hydrogen bonding. So the secondary structure of a protein is based on hydrogen bond interactions, which every amino acid has hydrogens that are attracted to more electronegative atoms of a different amino acid. And there's enzymes that actually will help fold it, either in folding over the top of each other or folding it into a helix. So this is referred to as an alpha helix or a beta pleated sheet. You just need to recognize, if we talk about an alpha helix or a beta pleated sheet, we are talking about those hydrogen bond interactions that lead to the secondary protein structure. So all proteins are going to have a secondary protein structure, and most polypeptides have a combination of alpha helix and beta pleated sheet within that structure. Now, the tertiary protein structure is now caused by all of these side chain interactions. So those different chemical properties of side chains lead to those nonpolar molecules turning in an aquatic solution, or the polar side chains coming out. If there's a sulfhydryl group, sulfhydryl likes to bond with other amino acids that have a sulfhydryl group. We've got ionic side chains that want to bond. So this tertiary structure is all due to these side chain interactions. And for some proteins, this is where the structure stops. So with a tertiary protein, it was one gene that coded for one polypeptide that folded into then this tertiary protein. However, there are some proteins in organisms that actually have multiple genes that all then form this conglomerate of a quaternary protein structure where it's two or more tertiary structures that are now grouping together to serve one function. Hemoglobin is actually an example in animals that actually is a quaternary protein structure. It's actually two different genes, and those two different genes and those two different tertiary protein structures form together to form that one functional hemoglobin protein. So that's my quaternary structure. So we do have to, and we're going to cover this multiple times, understand first of all the basics of a protein, where it's an amino acid is the monomer. They join together forming peptide bonds to give us a polypeptide chain. The sequence of those amino acids is the primary structure. The folding in an alpha helix or beta pleated sheet due to hydrogen bonding is the secondary structure of a protein. And then the side chain interactions lead to a tertiary protein structure. And then some proteins that are two or more tertiary structures forming one solid protein, that's the quaternary protein structure. So you will have to know those different structural elements of a protein. Now, let's talk about nucleic acids. Your nucleic acids are your DNA and your RNA. So you've learned lots about DNA and RNA, and with that, you know DNA is a double helix. You probably learned GCAT. So G, C, A, and T. We know that with this double helix of DNA, we know that these are the bases that are forming in the middle. But at this point, we need to make sure that we're stepping it up a notch and we're understanding more about that particular structure. So first of all, you absolutely need to know that for a nucleic acid, whether we're talking about DNA or RNA, nucleotides are the monomer. So a nucleotide is the monomer, and a nucleotide consists of three parts, and you have to know those three parts. So when we talk about G, C, A, and T, and then sometimes U, we're talking about RNA, well, that's the nitrogenous base piece. But we have to know that the side of DNA, so this backbone of a DNA or an RNA molecule is actually a pentose sugar. So in this case, if we're talking about DNA, it's deoxyribose and a phosphate group. So my DNA side chains, oftentimes in models, they don't show you the phosphate and the sugar. They're just showing you the difference in the nitrogenous bases because they're assuming that you recognize the sugar phosphate backbone that creates the side. Now then, on this nucleotide, so with this monomer, I've got my sugar, phosphate, sugar, phosphate, sugar, phosphate attached to the pentose sugar, then is a nitrogenous base. And nitrogenous bases then have these sections here, so these portions on them that want a hydrogen bond with another nitrogenous base. So this is what forms that double helix of, and base pairing rules of thymine always bonding to adenine, because thymine wants to make two hydrogen bonds, adenine also wants to make two hydrogen bonds, guanine wants to make three hydrogen bonds, cytosine also wants to make three hydrogen bonds. And this is what gives us our base pairing rules. So even when we're doing transcription and translation, it is these hydrogen bond interactions with complementary bases that actually allow for that to happen. So remember a nucleotide, the three parts to a nucleotide are phosphate, a pentose sugar, and a nitrogenous base. Now, some differences that you need to know though with DNA versus RNA is the difference in the pentose sugar. So in DNA, the pentose sugar, and we call it a pentose sugar because it's a 5-carbon sugar, and it looks like this little house with the oxygen right on top as a little chimney. So you'll recognize this structure. So the pentose sugar is either ribose or deoxyribose. And actually, if you look at this ribose structure here, there's two hydroxyl groups that are down at the bottom of ribose. But on deoxyribose, there's an oxygen removed. Now, that's really important in the stability of this molecule. Ribose is not a molecule that's very stable. It's going to last about 24 to 48 hours in an aquatic solution at room temperature or even at human body temperature. By removing this oxygen, this makes for a very, very stable molecule. So this is why we can find DNA segments in fossils that are 100,000 years old. They're still DNA intact. There's not going to be RNA still intact, though. Now, Know that it's a difference in the pentose sugar. And then the other difference is in the nitrogenous bases. In DNA, we have GCAT, G, C, A, and T. So there's this base thymine. But in RNA, there's no thymine, there's uracil instead. So we have G, C, A, and U. It's a substitution is what ends up happening. So then these are our other nitrogenous bases down here that are shared between DNA and RNA. And then the other difference that you're clearly seeing between DNA and RNA, DNA forms this double helix. It's actually two polynucleotides that then join together with hydrogen bonds in the middle. RNA is just a single strand. Now, there's other RNAs, we'll talk about those. They fold together, they form enzymes. RNAs, there's a ton of RNAs. So far, you've probably only been exposed to 3 different types of RNAs. There's a whole bunch more that have specific jobs in cells and organisms. Now, you will have to also make sure that you are recognizing with nitrogenous bases. So this is the AP bio level. You'll have to recognize again over here, these are my pentose sugars. Here's ribose, here's deoxyribose, and the difference is this loss of oxygen. That's why it's deoxy. You will have to make sure that you are understanding which nitrogenous bases are purines and which ones are classified as pyrimidines. So my purines and my pyrimidines, the difference is whether it's a double ring structure or whether it's a single ring structure. So we have to understand that in DNA actually ends up being this anti-parallel even distance between my two sugar phosphate side chains or sugar phosphate backbones. And so these two bases that come together actually have to be equidistant, but one is bigger than the other. So we need to know that purines have a double ring structure. These are my bigger nitrogenous bases. My pyrimidines are a single ring structure, and these are then smaller. Now, you will not have to, for AP Bio, have to ever recognize that this is adenine and this is guanine, but you will have to recognize the double ring structure and recognize that's purine. The single ring structure, again, you will not have to look at the structure and go, oh yeah, that's uracil. I can definitely tell that's uracil. No, you don't have to do that. You just have to look and go, oh, nitrogenous base is a single ring structure. That is a pyrimidine. So make sure that you are looking at that. Now, the last thing that we need to make sure that we are identifying, so this is again new for you that you didn't cover in your freshman biology class, is we have to understand that nucleic acids have direction. And when we start reading DNA, when we start doing transcription, translation, when we start talking about how replication happens, enzymes read DNA in certain directions. So we have to learn the directionality of our nucleic acids. So you have to understand the 5' end versus the 3' end of our nucleic acids. Now remember that I have a sugar phosphate side chain. Well, the other side is a sugar phosphate side chain, but remember that DNA, one of the things with DNA, in order for them to fit together, DNA is actually anti-parallel. So it is a double helix, two sides bound together by hydrogen bonds in the middle using complementary base pairing rules, but the other side is upside down compared to the first. So this is my cheerleader move of anti-parallel. This would be parallel, this is anti-parallel. Nucleic acids are always anti-parallel. So with that, we have to reference the 5' end versus the 3' end. Now, at the top of DNA, I have a 5' end, and here's the easy cheater trick. The 5' end always ends in a phosphate, phosphate 5, F and F. So remember that where there's the phosphate end, that's my 5' end. The other side is just going to be this hydroxyl group that's sticking out from my pentose sugar. This is the 3' end. So be able to recognize the 5' end versus the 3' end. 5' end is going to have the phosphate sticking out. 3' end is the side that just has the hydroxyl. Where those numbers come from is which of the carbons on the pentose sugar is this functional group attached to. And this is not something College Board needs you guys to know, but the hydroxyl group is stuck to the 3. It's covalently bonded to the 3rd carbon of the pentose sugar, and the phosphate is covalently bonded to the 5th carbon of that pentose sugar. And so that's where those terms 3' versus 5' come from. But again, for us, cheater version, phosphate 5 is what you guys need to know. So again, here are my 5' ends and 3' ends. Here's my double helix, my anti-parallel DNA showing a polynucleotide here, ending in the 5' end up top, but being anti-parallel, the 5' end at the other side then ends down at the bottom where the phosphate is. So with that, you guys have this worksheet to just summarize that information. So that is a Google assignment. So when you open that, it'll save it as your copy. And then that should be submitted as you summarize proteins and nucleic acids. So hopefully this helped for those of you who were picking up textbooks and missed today's lecture. So with that, email me with any questions that you may have, and hopefully I explained it clearly for you. So talk to you guys later. Bye. Let's get out of here. Exit. Let's get this, stop sharing.